Hi friends, recently a question was asked about dideoxynucleotide. You all know that pentose sugar, ribose sugar is a pentose sugar. It is having 5 carbon and 4 OH. If I write here HOH, HOH means there are OH group at C2, C3, C5 and C1. Okay, this is ribose sugar. But we found that in deoxyribose sugar, this oxygen is not there. If oxygen is removed at carbon number 2, oxygen is removed, this is called deoxyribose. So main difference between DNA and RNA is sugar. In DNA there is deoxyribose sugar, RNA there is ribose sugar. Question is asked, DNA is DNA, RNA. Yes, ATGC, AUGC. RNA is usually single stranded, DNA is double stranded, and A bind with T, G bind with C, A bind with T with 2 hydrogen bond, G bind with C with 3 hydrogen bond. Yes, so one difference, one very important difference between DNA and RNA is DNA is usually double stranded, RNA is usually single stranded. In DNA there is T and RNA there is U. But question was asked, what is the main difference? Main difference is not nucleotide or base. The main difference is sugar. Main difference is sugar. Okay, nucleotide, I will remove my this word nucleotide, because if we talk about nucleotide, there are possibility they will not go at this level. Simply remember base and sugar, which is more important. Sugar is important. Now I know that if I remove oxygen at C2, it is deoxyribose. If I remove oxygen from C3 also, this become dideoxyribose. This question was asked. I draw it here. Suppose this is pentose sugar, C1, C2, C3, C4, C5. C5 is having HOH, C3 is having HOH, C2 I don't mention. We found that C1 bind with base. Suppose A. Another pentose sugar, I draw another pentose sugar, C1, C2, C3, C4, C5. Yes, C5. C5 is having HOH. This is OH, this is OH. This is base G. Now, we all know hydroxyl group has tendency to attach with phosphate. So a phosphate group will come, a phosphate will group come and will make phosphodiester bond with these two hydroxyl group, and this is base plus sugar nucleoside. Base plus sugar plus phosphate nucleotide. So base plus sugar plus phosphate, base plus sugar plus phosphate. This is a nucleotide sequence. In this, A adenine guanine is there. If I ask you this strand is of DNA or RNA, you will ask me, sir, what is the situation of C2? If C2 is having OH, the sugar is ribose. And if sugar, the C2 is having only H, no oxygen, then it become deoxyribose. So if sugar is deoxyribose, this is DNA. If sugar is ribose, this is RNA. So you determine DNA or RNA on the basis of base or sugar. Sugar. Yes, this is OH free. This is called 5' end. This 3 OH is free. This is called 3' end. So every DNA or RNA strand is having 5' and 3' and we have read many times that we always read in 5 to 3 direction. Synthesis always occur in 5 to 3 direction. And question was asked, this is backbone of DNA or RNA. This is stabilized by phosphodiester bond. So backbone of DNA and RNA is stabilized by phosphodiester bond. Now DNA synthesis always occur in 5 to 3 direction, means if you want to elongate this DNA, a new nucleotide will be joined at this position. Suppose I take C1, C2, C3, C4, C5. Okay, C5 is having H, OH, C3 is having H. I don't mention OH, only H is there. At C2 and C3, only H is there. Means no OH group at C2, no OH group at C3. And you know, if no 2 OH group, dideoxyribose sugar, and this is base. Suppose C. So this is base, sugar, phosphate. So question was asked that if you add dideoxynucleotide in growing DNA chain is amplifying and you added dideoxynucleotide. In this dideoxynucleotide, there is OH group at C5 but no OH group at C3. When OH group of C5 is there, this OH group of C5 will bind with OH group of C3 to make phosphodiester bond, and it will not allow next nucleotide to join. Means dideoxynucleotide join itself but not allow next nucleotide to join because there is no OH group at C3 to make phosphodiester bond. This question was asked. This technique in which dideoxynucleotide is used, that is called chain termination method. Chain termination method. Okay, total number of human genome, yes, 20 to 25,000. 25,000. Number of genes are decreasing. Yes, it's not actually decreasing because the reason is that we are recognizing that one gene can give rise to more than one protein. That's why number of gene are decreasing. At this time it is considered 20 to 25,000 of gene. Now, before moving to purine catabolism, you must have an idea about DNA synthesis and at least DNA synthesis. Yes, DNA replication in prokaryotes. Yes, I can write here in prokaryotes. There are DNA polymerase 1, DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. While in eukaryotes there is DNA polymerase, DNA polymerase alpha, beta, gamma, epsilon, delta. Okay. So polymerase synthesize DNA, and DNA synthesis always occur in which direction? 5 to 3 direction. So all polymerase, yes, if I talk about polymerase, DNA polymerase, all polymerase have 5 to 3 polymerase activity. Yes, definitely question is asked. Yes, DNA polymerase 1, 2, 3 all have 5 to 3 polymerase activity. What is exonuclease? If any enzyme cut from the end, DNA or RNA is cut from the end, that is called exonuclease. If a nuclease come and cut from this side, 5 to 3 exonuclease means cutting from this side and moving in this side, 5 to 3 exonuclease. Exonuclease. Okay. If a cutting from 3 side to 5 side, then it is called 3 to 5 exonuclease. So we found that each and every polymerase in prokaryote is having proofreading. Proofreading means 3 to 5 exonuclease activity. Yes, proofreading means 3 to 5 exonuclease activity. Mug it up. All polymerase have proofreading activity except alpha and beta polymerase of eukaryote. No proofreading. Question. Clear? DNA polymerase 1 remove primer remove primer by 5 to 3 exonuclease activity. So you can say DNA polymerase 1, 2, 3 all are having, yes, these two activity. 5 to 3 polymerase activity, 3 to 5 exonuclease activity, but DNA polymerase 1 has one extra activity, that is 5 to 3 exonuclease activity. So I draw like this. This is DNA polymerase 3. Why I'm taking little bit more time, because recently asked question. It is DNA polymerase 1. This is DNA polymerase 1 is having 3 activity. Those activity are 5 to 3 polymerase activity. You all know all polymerase have 5 to 3 polymerase activity. All have proofreading, so 3 to 5 exonuclease activity, and this DNA polymerase 1 has extra activity 5 to 3 exonuclease activity. We found that in this DNA polymerase, if I remove this activity, if I remove this 5 to 3 exonuclease activity, this is DNA polymerase 1, and if I remove this activity, the remaining fragment is called clean out fragment. This question was asked. So clean out fragment is DNA polymerase 1 minus 5 to 3 exonuclease activity. 5 to 3 exonuclease is also called, activity is also called primer removal activity of DNA polymerase 1. Got the point? I just told you all polymerase have proofreading except alpha and beta polymerase. Now I will write gamma. Ma means mitochondria. Gamma polymerase synthesize mitochondrial DNA synthesis. Epsilon. E, epsilon looks like E. Leading, leading strand synthesis. Delta, lagging strand synthesis, lagging strand synthesis. So epsilon leading, delta lagging strand synthesis. Okay, so you will remember this polymerase. Now, purine catabolism, you all know purine metabolized to uric acid, and product of purine catabolism is uric acid. And we found that in this metabolism too, yes, salvage pathway is there, which in which enzyme is HGPRT, hypoxanthine guanine phosphoribosyl transferase. If this enzyme is completely deficient, then it is called Leishman syndrome. It is called Leishman syndrome. In Leishman syndrome, you will find the patient is having gout and there is self mutilation. There is self mutilation. Okay, while yes, many times you will find clinical question. They will give you multiple symptoms of gout, and they will show you a picture that a child is cutting his hand, a ear. So that is means self mutilation. And they will ask what is the enzyme deficient. The answer would be HGPRT. Yes, HGPRT is deficient. Partial deficiency is called Calais-Shagmiller syndrome. Calais-Shagmiller syndrome. Only and only gout is there. No CNS abnormality. Okay. Now, after each replication, a small portion of DNA is lost. But nature is very wise. Nature kept telomere at the end of DNA. Means TTA, GGG, TTA, GGG multiple times. These telomeres are present at the end of linear chromosome, and we found that whenever cell will divide, DNA replication would occur. This small portion of DNA would be lost. This lost portion would be telomere. The lost portion would be telomere. Yes, so after each replication, telomere is lost. So in somatic cells, what I said, in somatic cells, if I take example, somatic cell is having suppose 40 telomere. Somatic cell has 40 telomeres, and after each replication, one telomere is lost. So after 40 division, all 40 telomere will be lost, and if cell want to divide further, or if cell divides further, then the DNA will be lost. And if DNA is lost, that will lead to, yes, if DNA is lost, then it will lead to mutation, cell death, cancer, anything may happen. That's why somatic cell has limit to divide. That is called Hayflick limit. Question. Hayflick limit. Okay, question, depending on the number of telomere. But cancer cell divide rapidly and don't die because cancer cell have enzyme telomerase activity. Telomerase activity. Yes, cancer cell and germ cell. This telomerase synthesized telomer. This telomerase synthesized telomer. What is the composition of telomerase? Point it out. This is enzyme telomerase having two component. One is protein, second is RNA template. What happened? This telomerase protein protein do synthesis is having enzymatic activity, and this RNA is template. So enzymatic activity is present in RNA or protein? Yes, it is present in protein. And you know that this is not ribozyme. This is not ribozyme. Ribozyme are RNA having enzymatic property, but telomerase is not ribozyme. The enzymatic activity is present in protein. Now this telomerase synthesize sorry, telomer RNA act as a template. So this enzyme synthesize DNA from RNA. So any enzyme which synthesize DNA from RNA, that is called reverse transcriptase. Reverse transcriptase or RNA dependent DNA polymerase. RNA dependent DNA polymerase. So telomerase are complexes of DNA. Yes, in this DNA is there and protein is there that protect the linear end of chromosome. In most of the normal human somatic cell, telomeres shorten with each division. Yes, I told you after each division, a small portion of DNA is lost, and the lost portion is usually telomere. In stem cell and cancer cell, telomeric length is maintained by telomerase. Telomerase is a ribonucleoprotein. Yes, RNA template is there and protein is there. So you can say this is a ribonucleoprotein. It provides both RNA, it provides RNA template and polymerase. It also provides polymerase for the synthesis of telomere. This statement is true. The RNA of telomerase serves as a primer. No, this is not serving as a primer. This is serving as template. Okay, the polymerase of telomerase is DNA directed DNA polymerase. No, this is RNA directed DNA polymerase, reverse transcriptase. So false. Shorter 3-5 strand get extended. No, synthesis always occur in which direction? 5 to 3 direction. Now, base sequence of coding strand of DNA used for transcription has base sequence GTG. Look at me, this is double stranded DNA. Yes, the upper strand is suppose coding strand, the lower strand is template strand. Coding strand gave order to template strand go and make messenger RNA just like me. So this coding strand gave order to template strand go and make messenger RNA just like me. This template strand will separate, will synthesize messenger RNA. It will keep in mind the messenger RNA should be just like coding strand. But in RNA, you can't keep T. So it will replace T with U. So messenger RNA is just like coding strand except T is replaced by U. So now in this question, they have given coding strand of DNA GTC. GTG. What is the messenger RNA? Yes, you all know messenger RNA is just like coding strand except T is replaced by U. So in this GUG. So if question is given and coding strand is given, this is the simplest question. Means you will write the same sequence except T will be replaced by U. But in the same question, if I say the code, this is template strand, they gave this is template strand, what will be the sequence of messenger RNA? Now it will act as template, or you can say it is acting as template. Now this is 5 and this is 3 and this is 5, G2C, T2A, G2C, 3 and CAC, CAC. So point it out. If coding strand, the answer is GUG, but if they gave this is template strand, the answer would be CAC. Got the point? Now restriction endonuclease. Restriction endonuclease cut palindrome region. Palindrome. First of all, you will remember palindrome is double stranded DNA. It is double stranded DNA, and in this double stranded DNA, if you read from 5 to 3 directions, sequence should be same. Many times in many standard textbook, even Lipin code, just like they will give you example REFER. It will give you confusion because in it is good example. If you read in this direction or this direction, these things is same. But When questions are asked on palindrome, first question you will remember palindrome is double stranded DNA. This is not a good example, because if I write this is a nucleotide sequence or DNA strand, and we want to cut this DNA. This is single strand. We have to make double strand. So whenever question is asked on restriction endonuclease, before solving question, you will convert single strand to double strand. If I converted this single strand to double strand, option A, AAGG, CCTT, okay, because we read always 5 to 3 direction. So what is palindrome? If you read from 5 to 3 direction in double stranded DNA, in both strand sequence should be same. So upper strand the first nucleotide is A, lower strand the first nucleotide is C. This can't be same, so this is not palindrome. Let me take example. Yes, if you remember this refer and you will look at this option D, you may directly jump into answer yes, TAAT. No, this is not palindrome, because if I write TAAT, ATTA, yes, this first is T, first is A, this is not palindrome. The right example is GGCC. Yes, this is GGCC, GGCC. Okay, in this double stranded DNA, first is G, here the first is G, second is G, second is G, third is C, third is C, fourth is C, fourth is C. So this is palindrome. Palindrome means double stranded DNA. If you read both strand in 5 to 3 directions, sequence should be same. So in this answer is C. Restriction endonuclease cut, palindrome reason. Okay. Now, you know that one gene, one protein theory. Previously, we used to say one gene give rise to one protein. Yes, but now we say one gene can give rise to more than one protein. So one gene, one protein theory has been discarded. A question is asked, ApoB48 and ApoB100 are synthesized from same gene. Yes, if you take a liver cell, if you look at the nucleus, you will find a gene in liver cell, you will find a gene, ApoB100 gene. Yes, this ApoB100 gene messenger RNA is formed. This messenger RNA, yes, suppose is having CAA. This synthesize a protein. This is ApoB100 protein. So I take simple example. Suppose I took a cell from liver of a person, and in this liver cell there was a gene, ApoB100 gene. What I did in this ApoB100 gene, okay, I did nothing. In fact, I searched for yes, ApoB100 messenger RNA, and in fact, I found that this gene in liver cell synthesize messenger RNA. This is messenger RNA for ApoB100 gene. When this messenger RNA will synthesize protein, this will synthesize ApoB100. Nothing new, you all know. Yes, in intestine, you all know there is synthesis of chylomicron. Chylomicron. Chylomicron contain ApoB48. ApoB48 is protein, and protein is synthesized from gene. We search a lot but could not find gene for ApoB48. Suppose I take self of liver and intestine of same person, their genome would be same. Okay, liver cell is having ApoB100 gene, intestinal cell must have ApoB100 gene, but there was no separate gene for ApoB48. In intestine, the same ApoB100 gene synthesize messenger RNA, and you can say up to this point in intestine and liver cell everything is same. So if in this messenger RNA CAA is there, in this messenger RNA CA would be there at the same position. Yes. So in this intestine, if this messenger RNA synthesize a protein, it will synthesize which protein? Please comment in your mind. Yes, it must synthesize ApoB100, but we don't require in chylomicron in intestine, we require ApoB48. So what will happen? We found that in intestine there is an enzyme. The name of enzyme is cytosine deaminase. This question has been asked. Many times I have told you cytosine deaminase converts cytosine to uracil. Cytosine deaminase converts cytosine to uracil. Okay, so cytosine deaminase converts cytosine to uracil. Okay, now this becomes UAA. This becomes UAA. UAA stop codon. You all know UAA. You are away, you are gone, you go away, UGA. These are three stop codon. Okay, when this stop codon would come by this modification, chemical modification, this protein synthesis will stop at this point and a smaller protein will be formed. Means ApoB48 and ApoB100 are synthesized from same gene of ApoB100. The only method is by modification of cytosine to uracil. You edited the messenger RNA, and that's why this technique is called RNA editing or chemical modification. A question was asked that ApoB48 and ApoB100 are synthesized from same messenger RNA. Yes, it is synthesized from same messenger RNA due to answer was RNA editing. Okay, so one gene can give rise to more than one protein. ApoB100 gene can give rise to ApoB100 protein. ApoB100 gene give rise to ApoB48 protein. So one gene, one protein theory has been discarded. One gene can give rise to more than one protein. Question was asked, ApoB48 has approximately 48% mass of ApoB100. Before moving further, post-translational modification of messenger RNA, many time questions are asked. Modification are capping, attachment of cap at 5 end. What is cap? 7-methyl guanosine is cap attached at 5 end. Poly-A tail, attachment of poly-A tail at 3 end, yes, and removal of intron. Introns means intervening unit, just to support the genome. Yes, mitochondria, prokaryotes, no introns. Point it out. Intron means intervening sequence to support our genome. In eukaryotic genome is very large. To support our genome we have intron, and you will be surprised exon is only 1%. In few textbook 1.15%, 1% approximate. Question has been asked two times. One 2007, when this question was asked, at that time the value was 1.512, that a controversial value. Okay, at that time again answer was 1%. Even now the answer is exon is only 1%. Remaining part is intron. Most of the things are intron. Where intron is so much, intron is not going to take part in protein synthesis. Point it out. Intron is not going to take part in protein synthesis. Long time back a question was asked. They gave this DNA and they asked that in this DNA, this is intron, this is exon, this is intron, this is exon, and mutation occur at this point. Mutation occur at this point. Due to this mutation, what would be the effect on protein? Yes, you can think. Yes, introns are not going to take part in protein synthesis. Protein would be normal. Sorry, protein would be normal. Okay, this question was asked. So mitochondria and prokaryotes are not having intron. That's why mutation rate is very high in mitochondrial DNA. And this, when protein synthesis take place, means this before protein synthesis, post-transcription modification occur. One is capping, second is poly-A tail, third is removal of intron. Because introns are not going to take part in protein synthesis, it must be removed, and this is removed by small nuclear RNA, snRNA. Question is asked, small nuclear RNA remove intron, and you know RNA is having enzymatic property. Any RNA having enzymatic activity is called ribozyme. Ribozyme. So small nuclear RNA are ribozyme. So capping poly-A tail. Yes. Now, which is true about histone acetylation? You all know histone proteins are having positive charge. DNA is having negative charge. So histone protein is having positive charge, DNA is having negative charge. Question was asked, histone protein is having which amino acid? Option A histidine, option B arginine. You all know arginine will be the better answer. Arginine is most basic. Arginine and lysine are more basic. Actually, histone protein is having arginine and lysine, not histidine. Don't get confused by the name. And arginine and lysine is having positive charge. Due to this arginine and lysine, histone proteins are having positive charge. DNA is having negative charge. Now look at me. Suppose this my pen is DNA. This is sorry, pen is histone protein, and my two fingers are double-stranded DNA. Double-stranded DNA is having negative charge. Histone is having positive charge. Due to opposite charge, it binds tightly. It binds tightly. Okay, so histone protein DNA packing is done by which of the following? Need 22 means they are asking very simple question. If you know the concept, you can easily solve question. The answer was histone. Yes, this was question was asked long time back, means 4 5 year back, 6 year back. And if you know you have read this question, you can solve. So whatever you have read, trust me, dear friends, if you apply those information in your exam, you will clear your exam very nicely. The only thing I want, you should have trust. Trust in your hard work. Whatever hard work you have done, yes, trust it. And you will clear your exam. Okay, I have told you in psychiatry also, when you are positive in a stressful situation, unconscious information can come at conscious level. So in the last phase of preparation, I know I am little bit late. Many times we do have so many obligation in our life. You will appreciate later part in your later part of your life, dear friends. So but at this time of preparation, I would say leave everything, because nobody can spare all time for study. If you have wasted some times, even then don't worry, because we know that every person is having so many commitments, so many problems in life. Okay, so everybody is having the same scenario, more or less. Everybody has studied in the same way, same time period. Okay, the only thing which will give you extra edge is your positive thinking. If you are positive, yes, unconscious information will come at conscious level. There would be no anxiety if you are positive, no anxiety, or you can say controllable anxiety, limited arousal, and you will give your best. Okay, so definitely we are here. We wish you all the best. Yes, other than that, your own confidence, wishes of your family, your parents, your relatives, your friends are always there, dear friends. And you should consider these things as your strength. Okay, and when you will develop this strength, trust me, in whole life you will remain positive. Being a psychiatrist, I have seen so many students doing DM. Yes, they become disappointed. I don't know why, but many times uh, it happens. Okay, so only thing I want from you is yes, whatever you have done, trust me, it is paid. It will be paid. Okay, sooner or later. It's my personal experience. Paid doesn't mean only and only money. Yes, invested. I will not say wasted. I invested some time of yours, but trust me, if you remain positive, yes, other than this academic thing, yes, your motivational things, sometimes your positive thinking, sometimes your confidence helps you more than this academic thing. Okay, the only thing I can say again, that I have experienced, whatever hard work you have done, it is paid. Yes, I will tell you my story later. Yes, this part of my life I have never touched, yes, in any lecture. But trust me, it is very, very motivating. I will tell you, not now, but trust me, whatever hard work you have done till now, yes, it will be paid. And this exam will be only a small installment of that payment. Okay, dear friends? Okay, belief system, trust me, and remaining 4 5 days, 3 days, yes, put your all efforts without any anxiety. Okay, if anything is left, it's okay, it doesn't matter. Okay, your concepts are present in your unconscious mind, subconscious mind, or you can say preconscious. They will help you. Okay, those concepts will help you. So histone protein is having positive charge, double stranded DNA is having negative charge. Due to opposite charge, they are tightly bound. Tightly bound means condensed form. Condensed form means heterochromatin. Yes, in our nucleus, if you take a nucleus of a cell, cell is not dividing. That genetic material is present in the form of chromatin. Chromatin may be of two types: darkly stained, that is called heterochromatin; lightly stained, that is called euchromatin. Okay, darkly stained heterochromatin, lightly stained euchromatin. Euchromatin is relaxed. Heterochromatin is tightly bound. Yes, heterochromatin is condensed, euchromatin is relaxed. If it is relaxed, yes, you can easily separate them and synthesize messenger RNA. Okay, I told you double stranded DNA, two strands separate and one strand act as template. So if there is no space in condensed form, it can't do transcription. So heterochromatin is transcriptionally active, transcriptionally inactive, inactive. Okay, while euchromatin is relaxed. When they are relaxed, yes, they are relaxed, then you can synthesize transcriptionally active. You know, at a particular time in a particular cell, only few genes are expressed, other genes are kept in inhibited state. Why? Nature is very wise. Nature knows that these genes can be used in later part of life. For the time being, they are not required. What nature did? Nature kept those genes in heterochromatin, tightly attached to histone protein. Heterochromatin is transcriptionally inactive. Heterochromatin is transcriptionally active. They will not synthesize messenger RNA. They will not make protein. You all know DNA can't do work. If DNA want to do work, it synthesize messenger RNA. Messenger RNA synthesize protein, and protein do the work. Yes, DNA is the master. Yes, RNA is messenger. Messenger RNA is messenger or manager. Okay, manager, and the protein is worker. Poor guy. Okay, the whole work is done by protein. DNA can't do function. Yes, if DNA want to do function, it will convert, it will synthesize messenger and protein do the function. It is a normal scenario. Exceptions are always there, but you will remember the concept. Okay, suppose there is a gene in heterochromatin. Adverse condition came. Yes, I take example, adverse condition came. In this cell, a gene is present in heterochromatin. We know that this gene is activated, synthesize messenger RNA, it synthesize protein. That protein can destroy this toxic stimulus, but it is present in heterochromatin. Heterochromatin is transcriptionally inactive. So what will happen, that when this toxic stimulus came, what happened, this gene is activated, means this heterochromatin is converted to euchromatin. How it is converted? Yes, you all know this double-stranded DNA is attached to histone, because histone is having positive charge, DNA is having negative charge. I took one acetate. Acetate is CH3COO minus. What I said? Acetate is CH3COO minus. I will take one acetate and will attach to histone. When I will attach to histone, positive charge of histone is lost. DNA, yes, DNA is having negative charge, histone is having positive charge, acetate is having negative charge. I did acetylation of histone, positive charge of histone is lost, and this double-stranded DNA who is having negative charge will dissociate. Yes, DNA is attached to histone because histone was having positive charge. Now what I did, I attach histone with acetyl group. Positive charge of histone is lost, DNA will relax, relaxed DNA is euchromatin, transcriptionally active. Transcriptionally active, synthesize messenger RNA, messenger RNA synthesize protein and destroy this toxic stimulus. This is the concept. Whenever we want to inhibit the gene again, what will we do? We will do deacetylation of histone, acetyl group removed, again positive charge, DNA will bind, heterochromatin. Okay, so you can convert heterochromatin to euchromatin, euchromatin to heterochromatin by acetylation and deacetylation of histone. This is the method of gene regulation by not touching the nucleotide sequence. You are doing all these things above the genetics, epigenetics. This is called epigenetics. Okay. Yes, one method is true about histone acetylation, increase euchromatin formation. Clear? Another part of epigenetics is modification of DNA. Means epigenetics means modification of DNA or histone, but there will be no change in nucleotide sequence. Nucleotide sequence is not changed. Suppose this is promoter area. A question is asked, promoter is attached with RNA polymerase. Okay, and we found that if we want to inhibit transcription, we can do methylation of DNA at promoter region. Which nucleotide is methylated? Cytosine. Yes, cytosine is methylated. Sometimes you will find that they will give cysteine. Cysteine is not nucleotide. It is, yes, no, it is not base. It is amino acid. So cytosine is methylated, and methylation of DNA at promoter region at cytosine, yes, decreased transcription. Decreased transcription. Clear? So these are common questions of epigenetics. Now, RNA interference. Yes, this has been asked for 4 years. Multiple question. Every exam is having questions. So I will write RNA interference or RNA induced silencing complex or silencing RNA. All are same. Okay, I am repeating again. RNA interference, silencing RNA, RNA induced silencing complex, okay, microRNA, small interfering RNA, small interfering RNA or RNA interference. For your question purpose are same. Let me give you a simple form. What happened? Suppose there is a cell. Yes, I am discussing RNA interference. Suppose there is a cell, a toxic stimulus came. In this cell, in nucleus, there was heterochromatin. This heterochromatin, if it is converted to euchromatin and synthesize protein, this protein can destroy this toxic stimulus. I told you we have so many genes which are kept in dormant state, inhibited state. When we require these becomes active by, yes, epigenetics. I told you, just like acetylation, deacetylation of histone. So this was the gene which was present in heterochromatin. Adverse condition came, and we know that this gene will be able to tolerate this toxic stimulus. Adverse condition, this gene is converted to euchromatin. Now this gene synthesize messenger RNA. This messenger RNA synthesize protein, and protein will destroy this toxic stimulus. I told you function is done by protein. Function is done by protein. DNA can't do function. Okay, most of time. Now, what happened? This toxic stimulus came, gene is activated, heterochromatin to euchromatin, synthesize messenger RNA. Messenger RNA has not synthesized protein till now. Meanwhile, this toxic stimulus is gone. Meanwhile, this toxic stimulus is gone. Now, is there any need of protein? When this toxic stimulus is gone, I don't want to synthesize protein from this messenger RNA. But yes, who will stop this messenger RNA? Who will stop this messenger RNA? Yes, that is silencing RNA. Yes, let me write here. We found that this toxic stimulus is gone. We don't require this protein. So how this messenger RNA is inhibited? We found that silencing RNA. Silencing RNA. Suppose this is messenger RNA. This is 5 end. This is 3 end. When we don't want this messenger RNA to synthesize protein, what will happen? Silencing RNA would come. I take different colors. Silencing RNA would come, or micro RNA would come and will bind to 3 end. Question was asked. Micro RNA bind to. Yes, three untranslated question. Okay, when it bind to three untranslated region, it will not allow this messenger RNA to synthesize protein, and definitely it is not required. Why questions are being asked? In many cancer, you know, there are proto-oncogenes. Proto-oncogenes become oncogenes and synthesize protein, and protein act as a growth factor, or you know better than me. Yes, and it produces a cancer. So if I know that this proto-oncogene is overexpressed, I can inhibit their messenger RNA by making microRNA for that messenger RNA. And in later part of, after 10, 20 years, you will find that for most of the treatment, microRNA would be used. Okay, that's the reason that microRNA question is asked. First question, microRNA bind to messenger RNA at which end? The answer is 3 untranslated region, and it will inhibit the protein synthesis, and this phenomena is called gene knockdown. Gene knockdown. Gene knockdown. So okay, how you will remember microRNA? MicroRNA, yes, bind to M. If you rotate once, it looks 3 untranslated region, and it causes gene knockdown. DOWN. So if you rotate once, it becomes 3. If you rotate once, it becomes W. Gene knock down. Gene knock down. Okay, got the point? See, okay, knock in, knock out. Sometimes you may get confused. So you will remember 3 W. Yes, knock down. DNA protein interaction can be studied by, yes, you all know steroid hormone receptors are having zinc finger interact with DNA, and this DNA protein interaction is mediated by, I usually make mnemonic like this. These are the foot of this duck or whatever you say, DNA footprinting. DNA protein interaction is mediated by DNA footprinting. Mnemonic snowdrop, SNOWDROP. Yes, southern blotting for DNA, northern blotting for RNA or cut, western blotting for protein. You have learned South India dosa, North India roti, yes, western pizza. Okay, so tumor cell, whichever one most appropriate for this purpose. So this was DNA, RNA, and protein. But whenever you find, yes, multiple mutation, compare gene expression in two different cells. Wherever you find the word global pattern of gene expression, you will find the answer microRNA. Sorry, microarray. Sorry for that. Microarray. Global pattern of gene expression. Yes, diagnosis. Yes, technique is microarray. This is DNA chip. Sometimes they may ask MCQ, picture MCQ. In this question, if even if you don't read this question, just remember technique listed below was most likely used to analyze RNA. For RNA analysis, which technique is used? Yes, RNA roti, no blot, no the blot. Enzyme inhibition may be competitive and non-competitive. I know that I have summarized all the topics in very small, you know, uh, points, but trust me, it will give you a confidence. Okay, ultimately I want you must be confident when you are going for exam. You have really really done hard work. Okay, just like I can understand even yes, it is around, let me check what is the time. It is, yes, why I'm telling you, yes, why I'm showing you all these things, what my purpose means for doctors. Yes, this is 12:30 around. Okay, so being a doctor, many times we have to study hard, but It is useful. Okay. Whosoever says that doctors are underpaid, I say yes, maybe. Yes, if you talk about the finance, okay, money. But dear friends, whatever you can do with the help of your knowledge, that would be, most rewarding than any money. Okay. So I want to share, but will not invest time in that. For the time being, yes, trust me, yes, you have really worked hard and it will be paid. Okay, with this confidence, if you have believe in me. Yes, most of you have in contact with me. Yes, for few 2 3 months I was not able to remain connected with my students so frequently. Okay, but I tried. Okay, so if you know me, I would say that believe in my words, and your confidence will increase in your exam. Okay, whatever hard work you have done, if you will remain cool and calm and will have confidence, okay, okay, this question they have twisted, let me search what may be the concept which can be used here, you will find the answer. I would not say that you will waste extra time, but after some time it would be a habit, because in exam we have arousal. Yes, Yerkes Johnson curve. So our activity is increased, our performance is increased. With this confidence, you will appear in exam. Okay. Competitive inhibition. In competitive inhibition, there is competition. Means competition occurs between two similar persons. So structural similarity is there. Structural similarity is there. They bind to the same active site. When they bind to active site, same active site, affinity decreased. If affinity decreased, Km is increased. Km is inversely proportional to affinity. V maximum remains same. So you will remember like this, V maximum remains same. Non-competitive, I usually remember this is a cartoon having a very heavy pittu bag, yes, very heavy bag. He would not be able to run very fast. V maximum is decreased. V max decreased. Non-competitive, yes, no change in Km. Non, I can write like this, non-competitive, competitive. So mnemonic noun competitive, I write this, no change in K, noun competitive. Yes. Now this is Michaelis mental theory and curve. In this you will find this is V max. So if you want to solve the question on this graph, two times recently question has been asked recently, but it before that 5 6 year back the same question was asked in aims. In this is V max. If I say if V maximum is not changed, if V maximum is not changed, V maximum remains same, that is competitive inhibition. So if they give graph, in this graph, if they give, you will say this is V max, V max is same. If V maximum is same, this is competitive or non-competitive? Competitive inhibition. Okay, if graph is there and V maximum is decreased, V maximum is decreased, yes, V maximum velocity, he can't run with maximum velocity, then it is non-competitive. Non-competitive. So simply remember V maximum, same, competitive, decreased, non-competitive. Okay. Now, line Weaver-Brook plot, this intersect, Y intersect is called 1 upon V max. So you will remember this, 1 upon V max. In competitive inhibition, no change in V max. So competitive inhibition, no change in 1 upon V max. No change in 1 upon V max. So if a graph is given and this is normal graph of enzyme, after inhibitor this is, so in both of these conditions this is V max, 1 upon V max. So 1 upon V maximum is same, 1 upon V max is same. Now V 1 upon V maximum is same, that is competitive inhibition. In non-competitive inhibition, V maximum is decreased, so 1 upon V max is 1 upon V max is increased. 1 upon V max is increased. Yes, 1 upon V max is increased. This is non-competitive. Non-competitive. 1 upon V max is non-competitive. Now you remember uncompetitive. I say don't remember anything. Just look at the graph and you will find u. You will find u. You can see yes it is hanged, but don't worry. You will find u. Uncompetitive. You will find u. Yes. In this there is no u. In this there is no u. In this there is no u. But in this clear cut u. Uncompetitive question has been asked. Uncompetitive, I invert u and I write km and v max. So km decreases, v max decreases, v max decreases. So km and v max both are decreased. That is uncompetitive inhibition. Okay, so this graph has been asked. Thank you for patience listening. Yes, best of luck. And I know that you will do your best and remain cool and calm. If you will remain cool and calm, your performance will increase. And again, I say your trust will help you a lot. Thank you.